All right, hello everyone. Welcome to this panel Q&A about paths to a PhD. I'm Maria, I'm a first year PhD student, and we have here three lovely other PhD students, so Dominika, Tess, and Paul. And yeah, without further ado, I'm gonna let them introduce themselves, show you a bit about their own projects, and then we'll get to questions.
So, hi, I'm Tess. So I'm doing my PhD. I'm in my third year at the moment, and I'm doing it with Leon Laniardo and Tom Barden, sort of 50-50. Is this the slideshow?
Yes.
Will I just click on? Yeah. So I'm in the third year of my PhD, so I'm sort of coming towards the end of it. And my project focuses on the visual system, particularly the very first synapse in the visual system pathway. So I'll give you like a brief overview of my project, and then we can go like into more detail about like the route that I took to get into this PhD. So like the labs are very interested in the visual system because all the the light information that comes in through the eye and hits the retina, that's what allows us to perceive like motion vision, allows us to perceive color, sharp edges, lots of things, and allows us to communicate with the world. And I'm particularly interested, so here you can see the photons hitting the retina, and I'm interested in the very first synapse here. So it hits the photoreceptors, which generate this like nice graded signal and transmit it to the downstream bipolar cells, and then the ganglion cells, which eventually send the signal back into the brain. Okay. So that's why we're sort of interested in studying the visual system. So this is the first synapse, and on the right-hand side, I've just got a schematic here as well of the first synapse. So in pink, we've just got the pedicle of the cone photoreceptor, which is just like the end of it. And it forms this really tight synapse with the postsynaptic horizontal cells and bipolar cells. And what I want to do here is I want to measure the output of the photoreceptor so I can see like how they're talking to the downstream cells, like what information gets transmitted, how much of it, because the dynamic range of information that we have here is going to impact what we have further down the line and like how we can perceive from the world around us as well. So to do this, I'm using a zebrafish model. I thought, oh yeah, here it is. Here's a little picture of it. I don't know how many of you are familiar with the zebrafish, but it's used in a lot of neuroscience research and a lot of like heart research as well, actually. But we use it in the visual system because the larvae are optically transparent. They're really easy to genetically manipulate. So they're a really nice model to look at retinal function in vivo. So we can look at it right in the live animal eye. And the zebrafish that I use, so they're expressing a glutamate sensor called glucifer or iGlucifer in the horizontal cells, which are postsynaptic to the photoreceptors. So every time a glutamate molecule hits the sensor, it will light up and it will give a signal. So it's kind of like an antenna, like telling me, okay, what's being released from the photoreceptor. So I put the live zebrafish under a two-photon microscope, which you guys might be familiar with as well. So it's just like very high resolution in terms of being temporally high resolution and spatial high resolution as well. And I can record the signals from an individual photoreceptor and see what neurotransmitter it's spitting out and how it's talking to the next cell down the line. And here in D, I've just got two examples of what those sorts of recordings look like. So I've got two different types of cells. So the top one is from a red sensitive cone photoreceptor. So you get these nice big transients, and then the green photoreceptor, color opponents, so they do the opposite thing. And what I'm finding from like my recordings is that there's certain cone types, and I'm particularly interested in the red cones because they're like an achromatic channel, and they're like extremely robust. They get very reliable responses, and they're like really important for basically staying in like black and white essentially. And I'm also backing this up with lots of like morphological analysis as well. So I'm using these sorts of things like immunos, which is what I've got up on the slide here, so I can look at the kind of gross morphology of the photoreceptors and how it changes in different regions of the eye. So I'll just quickly show you what we can see here. So this is a zebrafish retina, 6 days after the egg has been laid, and it's like of like half the length of like my little fingernail. They're absolutely tiny. So it's really great that we can get these kind of images. And different regions of the eye have got different specializations for their behaviour. So for example, you can probably tell here, this is called the acute zone or the strike zone, and it's quite thick. The photoreceptors are really big. They've got really big outer segments, which is what they use to catch the photons. And it's because they use that part of their eye to look up and catch prey. So they've got like the tiny little prey, so they need these really big photoreceptors to detect them. So it's that sort of thing as well. And yeah, that's an overview of my project in the Barden lab and Lanyardo lab, and happy to take questions now or at the end.
Yeah, it's related to the project.
Or the labs or anything.
Yeah, how does working like the 50-52 labs work?
Yeah, so I think, so basically when you do your PhD, you get 2 supervisors. And you'll always have a co-supervisor, and sometimes the split really changes. So it might be 90% or 10%, or it might be 70-30 or whatever. But mine's 50-50. And I really love that, because both my bosses have worked very closely together in the past, but they've also got quite different expertise. And I think at the beginning, like you kind of got to find your way to manage how that's going to work in terms of like how often you meet together. And all the rest of it, but that, like, it works very well for me. And they're 2 labs in the same building as well. So yeah, so that works. Yeah. Okay. Do you want to sit on the seat?
Sure. Maybe I'm a bit short, so I might just stand. Yeah, so hi everybody, I'm Dominika. I'm a second year PhD student in Chris Bird's lab. And I'm just going to give you a brief overview of what it is like to do a PhD in cognitive neuroscience. I'm actually in the School of Psychology, but we do research related to memory, and is not just the behavioral questions that we are investigating, but also questions related to the brain. So that's how the neuroscience a bit comes in. So yeah, the overarching question that we ask in our lab is how people remember experiences, events from their lives. And more specifically, we are interested in neuroscience questions like what are the brain systems that are involved in memory, and how each brain region is involved in different aspects of memory. And we are using neuroimaging techniques as well as neuropsychology to investigate this question. But we are also asking questions that are more psychological and related to the mind. So, for example, what are the units or the episodes of an experience that people perceive and recall? What constitutes an event in memory? So what are the features that define and separate different types of events? For example, how does your mind separate a grocery shopping event from an exercising event and create separate memories for these? And what makes an event memorable? So how come that you remember some events very well, but you don't remember some events that much, or that you forget entire events altogether? So when investigating these questions, we make hypotheses related to how we think that a memory works. And then we design experimental tasks to test these hypotheses. So in our lab, we ask people to do these tasks, and then we compare their behavior and brain activity between the different experimental conditions that we have. And we try to design stimuli that resembles naturalistic real-life experiences, but are well controlled, so that we make sure that what we are comparing are not confounded by other unwanted factors. So, for example, in one of my experiments, I was interested in when people are experiencing that an event has ended and that a new one has begun. And one theory says that people are experiencing changes in events when they experience a prediction error, when something unexpected happens that they didn't predict before. And so to investigate this, to test this theory, I created stop motion videos and I asked people to watch them. And in these stop motion videos, I had two scenes creating an unexpected change between two events. For example, a person just finishing doing yoga, and then after this scene, and the person making breakfast. And we also manipulated that in one scene, in one condition, there would be a surprising action. So for example, in this one, the person is just making cereal for breakfast, and the surprising version of the action, let me just go back here. The surprising version of the action is that the person is squeezing a sponge into the cereal bowl, whereas in the neutral condition, the person is pouring milk into the cereal bowl. So what we were comparing here is behaviorally, we tested whether people indicate that there was a change in event at these event boundaries. So when you change from the yoga to the breakfast making, as well as when the surprising action is happening. So is it the case that every time something unexpected happens, people perceive that there was a change in events? So that is a behavioral question, but we also wanted to test what are the neural correlates of perceiving that events change. So in my experiment, I used fMRI to test the brain behavioral relationship. So fMRI is measuring brain activity by detecting changes in the blood flow in the brain. And we assume that when there is more oxygenated blood in a given region at a given point, that region is being activated at that time. And so later, when we analyze our data, we can get these statistical maps that are showing that which brain regions were more activated in one condition compared to another. So for example, when people were perceiving the squeezing the sponge act compared to when they were perceiving the milk, pouring the milk action, these yellow areas were more activated. So fMRI is just one of the techniques that we can use, and it has good spatial resolution, although it doesn't have single cell resolution like the techniques that Tessa uses. And it also has bad temporal resolution. So depending on the question you are asking, you can use many different types of techniques. So some other popular techniques in cognitive neuroscience are, for example, EEG and MEG, if you are interested in the time course of electrical activity in the brain. But these are all correlational methods. If you want to see whether a brain area is necessary to perform a cognitive function, then you might want to use TMS that causes temporary lesions to the brain or to the specific brain area that you choose, or study patients with neurodegenerative diseases or lesions to their brain. More recently, there are labs that are also using intracranial EEG that gives actually the single cell resolution when studying living people processing information. So when they are going through surgery, they can plant electrodes into the brain and record electrical activity from single neurons. So yes, if you are, cognitive neuroscience is very, very diverse, and if you are interested in both behavior as well as how behavior or cognitive functions arise from brain activity, then cognitive neuroscience field might be for you. And yeah, I'm very happy to take any questions if you guys have. And also, you can visit our lab website if you want to know more specific details about our research. And thanks for listening to me. Do you have any questions now? Or?
How are the results so far?
For this project? I'm just analyzing the data now, and what our lab is doing is actually pre-registering all our studies. So we state upfront what our hypotheses are and exactly what results we are predicting. And so far it seems that some of our hypotheses are wrong. So it's, yeah, I don't mind as long as I'm not doing something incorrectly, and that's why we're not finding what we wanted to find. But that is just the way that science is, and sometimes you find things that are unexpected and not the way that you wanted them to go. So yeah, that's how the results are going for now. Yeah.
Okay, so I'm Paul Kinghorn. I'm on the Neuroscience PhD program. It's a 4-year program. The first year is rotations in different labs, and then you do your 3-year PhD. So I'm the 3rd year, so the 2nd year proper. I'm in informatics, although I would say the neuroscience program is sponsored by life sciences, psychology and cognitive sciences, medical school, and informatics. So you do your rotations, find yourself a supervisor, and then end up in your home school. And then I would say in informatics, we have a lot of people working on, let's call it brain stuff rather than neuroscience. But we have mathematicians, philosophers, real expert coders, all sorts of people. I would say I'm a jack of all trades, master of none. My original degree 35 years ago was in maths, and I worked for almost 35 years. Then I did a master's in intelligent and adaptive systems here, and then on to the PhD program. So what I look at is, or let's say the motivation here, is that there are 2 types of behaviour, or generally thought to be 2 types of behaviour. There's fast, habitual behaviour and slow, goal-directed behaviour where we think about things. So say reflexive, habitual behaviour and reflective, and Daniel Kahneman wrote a book about it. It's very famous, Thinking Fast and Slow. Also, Stanskovich and West came up 20 years ago with the idea of System 1 and System 2 thinking. So from a cognitive sciences point of view, that's the motivation. Then in the world of machine learning, if we think of reinforcement learning, there are 2 distinct types of reinforcement learning. One is model-free, where you just learn a direct mapping between something happens and I do it. So it's very like habitual behaviour in cognitive sciences. And model-based, where something happens, I work out what it means, and then I come up with an action in response. So that's the goal-based. And, if you read a lot of papers, the idea of model-free and model-based are just intertwined with habitual and goal-based. They tend to be the same thing. So the famous reinforcement learning book is by Sutton and Bartow. It's real machine learning stuff, but then they talk about habitual and goal-based behaviour and how this could replicate it. So that's cognitive sciences, machine learning. Then if you look at the world of neuroscience, there is evidence that different parts of the brain cause habitual behaviour, and two parts of the brain which cause goal-based behaviour. And lesion studies on animals and fMRI studies on humans suggest that different parts of the striatum, for example, different parts of the prefrontal cortex are involved in these two types of behaviour. But there is also quite a lot of studies which says, oh no, it's not, it's all mixed together and it's all one system, and this model-free versus model-based isn't the right way to separate it. It's more of a continuum. It's challenging the notion of separate model-free and model-based learners. So my PhD, as I say, I'm a mathematician that does a lot of coding, is using something called hierarchical predictive coding, which is, I'll explain a little bit more in a second, but it's the hypothesis that a single system can produce both habitual behaviour and goal-based behaviour without it being two separate systems, and justifying this idea that it's a continuum. And you may have heard of the Bayesian brain. So it's driven by the idea of the Bayesian brain, that the brain predicts what's going on and we have a response. It sort of infers what's going on from its predictions, combining its predictions and the outside world. The free energy principle is something that's very heavily involved in this with active inference, which I'm not going to go into detail here. But it leads to the idea of hierarchical predictive coding. So if you look at this picture here, with a sensory input at the bottom, generating an action or a muscle, that could either go all the way up the nodes with lots of processing going on before you end up with an action at the bottom, or it could be a much more fast come up just to the first node and then straight back out. And the hierarchical predictive coding model tries to explain that, how you can either use all the system or just use some of the system, depending on the precision of your beliefs. And so there's a lot of mathematical modeling trying to prove that. So what do I do all day? Well, I program an awful lot of predictive coding models in Python mainly, trying to show how they react to different situations. So it's very theoretical. So it's very half my time doing that, and the other half of my time I spend reading papers, and a lot of papers, a lot of maths, even more machine learning papers, because an awful lot of the machine learning people these days are actually very inspired by, well, there's two types. Some people inspired by the brain, trying to develop machine learning techniques, but even more people are more like what I'm doing, which is trying to use machine learning to explain what's going on in the brain. Some experimental cognitive science papers, because I was looking for results that we can try and then compare that with what does our model predict. Quite a lot of philosophy, as I was saying a minute ago. We have Andy Clark, who's a philosopher in the informatics department, for example, well, world-renowned, very big in the idea of predictive processing. He's not in the line, so I'll say, but he's never shown it. He's got these ideas, but what we're trying to do is show actually in a model, well, this is actually how it would be implemented, and try and tie that up with some cognitive science data and with some neuroscience data. So it wouldn't be fMRI, it wouldn't be EEG, it would be much more like stuff Tess looks at, at the neuron by neuron level. So at the moment I'm talking to somebody over in life sciences, Miguel Maraval, who has quite a lot of data on the mouse spiral cortex, and seeing if we can try and use that data and see if the representations it comes up with are the same sort of representations our computer model comes up with. Yeah, so in summary of my hypothesis, here's a fly. A fly gets sensory input in and quickly reacts with a muscle, it's reflex, or it's just optic flow, it's following a certain simple rule. A human being in the right-hand side might use a lot more of the brain, but when you learn to do optic flow, the famous fly ball in baseball, if somebody hits the ball, there are very specific rules about how you catch it, and you do it without knowing it. But if you're good at catching balls, you will follow those rules without thinking about it. So you move from this model-based, working out what's going on, to instinctive, it's all in one system. So that's what I do. Much less experimental than Tess, Dominica. So it's very easy to sit about all day, and you get to Friday and you find out you haven't moved forward again because you don't have a specific plan. You have to be quite disciplined in terms of, well, what am I trying to do at the moment? How am I going to do it? And keeping on top of it, whereas these guys presumably have a 3-year plan. I'm going to do this for a few months, then I'm going to do this. I'll make it up.
And they get rewritten every 3 months.
Yeah, but you know, it's with mine. It's more, well, let's piss about with this stuff and see what happens. So you got to be disciplined, you know. Any questions on that? Yeah.
Yeah, so it's clearly quite an interdisciplinary area. So there's a lot of bits of knowledge and expertise that are needed to go into it. Presumably, if I were to go into a PhD in this, I would be expected to know a lot about each of those already. So what's the sort of process like, getting up to speed in this area?
Well, you're doing a master's in neuroscience, is that right?
Yeah.
Yeah. So, I mean, I would say one very good thing that I did, and I did it deliberately, was go on the neuroscience program, because then I went, the first year you do 3 rotations. So I did rotations over in life sciences, learning about experimental techniques in the mice, and you know, the sort of what's possible, what's not. I did my master's in adaptive systems, which was very where maths meets machine learning meets philosophy. So in my first year of the PhD, I took some neuroscience stuff where I learned what a protein is, because I've got no idea what a protein is, you know, what, these sorts of things, you know, that was the bit that I was missing. So identifying what courses are going to get you up to the level you need to be at. You're not going to be expert in everything, you know, so learn as much as you need to learn, I would say, and, you know, for you to be able to do the stuff you want to do. And talk to people. So as I say, I'm in informatics, but at the moment talking with Miguel, who's over in life sciences, about what he's doing and what data he has and what ideas he has and how we can marry them together.
Right.
Do you have any questions online? No, that's just.
Yeah. Right. I guess I'll stop the screen share.
Oh, sorry, I was going to say, I've got
Yeah.
a general question.
Yep.
I wasn't sure if we were doing that.
No, yeah, go on. Yeah, sorry. This is more about how to get into a PhD. Or
Yeah,
what,
just kind of for everyone. How did you kind of decide, and how hard was it, the kind of process of kind of settling on what you're going to do and kind of what you're going to research in your PhD?
Well, mine started off woolly, you know, and so during my masters, I had certainly a belief and interest in predictive coding, the energy principles. So I'd done a lot of reading at Royal Andy Clark and from the mathematical side of it, Carl Friston. But coming back to your point, didn't understand half of it because it is a notoriously difficult subject. Still don't, but you know, I understand enough to do what I need to be doing. And then when I started with supervisor, I had a sort of general idea about the sort of area, habitual versus goal-directed. During the first year, really developing that idea into a sound hypothesis and what the model would look like, and what might be a crackpot idea and what is actually a reasonable idea. So certainly the sort of path that I took, I didn't have to turn up with that. Here's a 3-year plan, here's what is going to happen, and I'm going to arrive and next week I'll be doing this experiment and this experiment. It's much more in the early days finding your way. But you have to be a bit more specific than I want to investigate the brain.
Yeah.
What do you guys think?
So the project, when I applied for mine, it was a predetermined project, and I think that is often the case in pure neuroscience and probably other departments as well. But it was sort of the kind of the questions were sort of predetermined, and then as I got into it, like, you know, obviously things change and you realise, oh, I actually can't answer that question, I need to sort of change it or whatever. But yeah, it was sort of like a very sort of probably more specific than what Paul's was originally, and that was just based on what I've been interested in.
For me, when I was doing my undergraduate and master studies, I tried to gain experience in different labs, and I also did like a research assistant job for a year before I went for a PhD to really think about what sort of area interests me the most. And when I applied here, I was looking at lab. Well, when I decided that I want to do something related to memory, I looked around for labs that are looking for students as well, because sometimes you really want to go somewhere, but it's impossible because there's no space. And also, I came from a lab where they are using quite standard psychological tests, like just showing individual words or pictures. And what was another aspect in coming here is that here the lab is using very naturalistic stimuli. So sometimes you might also want to think about what sort of techniques you want to explore in your research.
So we have a question in the chat. I guess this applies more to you, perhaps. Did you have to look for funding, or Brexit hadn't impacted on your applications yet?
Well, I did my undergraduate in the UK, so Brexit didn't affect me because I have been here long enough. But I did. So I am funded by the School of Psychology. So I applied specifically for this funded position, but I didn't look elsewhere for external sources of funding. So I'm afraid I can't help with that.
I'm funded by the Neuroscience program. I was also about to apply for the Lever program, which also funds people in informatics and in life sciences, if you like. And they are both programs that take several people each year. And then there are also people in our labs who are funded directly through funding they fund themselves, and others, I guess, in those as well.
Yeah. Yeah. Do we have any? Well, not in our lab, but Marina is self-funded. Yeah.
So I think there are. I don't know about the Brexit question, to be honest. I don't know what the situation is with neuroscience and everything. No, but actually, we've just brought Italian people in this year, so I think the Brexit thing is not an issue.
Rachel, there's actually a fund you can apply for, which is specifically for scientists who want to move to the UK. I don't know if you know more about this or not.
Yeah, also because I did my undergrad here as well. I don't have any issues with that, but I do think they do take some international students.
Yeah, I know in the neuroscience program this year there are 4 people. One's English, one's Italian, and I think come straight from Italy.
Right, okay.
So Ruth Starr has to be able to answer on the neuroscience program,
Yes.
and Lever, who I guess undergrad in the Philippines as well.
On just another point on like the bit of funding, I think like I've spoken to you about this before, like if there's a lab that you're interested in going to work in, it's really worthwhile contacting the lab directly, because even if they don't have a position, advertisers available there and then, 9 times out of 10 there's going to be ways and means to get funding, whether it be that you have to wait a year and like work as a research assistant for a little bit and then you get your funding, which is what I did, or there's often different ways to do it, or join up with another lab as well. So that's just something just because it's not advertised doesn't mean it's not possible.
There's some more questions here. So what coding software do you use, and how long does it take for you to feel confident with it?
Well, mine is I spend an awful lot of time coding, and it's Python, with various libraries, some off the shelf, some that we've developed ourselves. How long did it take to go on confident? Well, I'd never coded until my master's, so during my master's I was doing quite a lot of coding, and so most things, the more you do it, the better you get, really. So, I mean, I don't know, Amy, what sort of area you're looking to get into, whether it would be something that involves heavy coding or something that uses less. But I mean, I guess these guys still have to be able to do coding.
Yeah.
I have to code up my experiments and the analysis, so it is, and even for psychology, there is a lot of coding involved.
Yeah, I think it's becoming increasingly more necessary skill to have, and like it will just stand you in so much better stead if you do have like some kind of coding experience or whatever. And there are so many free resources available online. There's something called Code Academy, which I started off with, and they've got some paid courses. You don't have to pay for it, just do the free ones. And that was really nice. It kind of like opened the box to coding, which previously just seemed like, you know, something only like hyper intelligent people would even know what it is. So yeah, it's like I would just start taking a look at that, and then you know, I don't know, it's just nice in your interviews, isn't it? You can say like, yeah, I started looking at like, you know, different languages and all that sort of stuff. Just have some kind of awareness.
So Amy says she's neuroscience. I know a few people in Python, but I've only used R before. So R, yeah, you might use that for the stats, but Python or MATLAB, there's lots online you can get on with. And there are actually courses the university runs now, I think. I know they were doing one a few months ago, 2-week, 4-hour-a-day Python course, you know. So you can look out for that if you're going to be. So even if you were, say you're starting a PhD here, you know, during your first term of it, you can be learning Python.
Yeah, so that's what I did actually in my first week. I did this kind of intense 2-week,
Right, yes.
4-hour thing.
Yeah. And also, you know, you will do, or you have the opportunity to audit any master's course that's been taught. So an awful lot of people sort of forced into the maths course, I think, you know, which I'm teaching assistant on, which is like it's master's maths, but it's not master's level, but it's maths for masters and PhD neuroscientists, data scientists. So things like that, you know, you'll feel, well, going back to your question, I might have a gap in my knowledge. That maths course is a gap that an awful lot of people have. And so their supervisors tend to push them into that. Whereas I've got maths background, so I was doing synapse type courses.
What are the assessments like during the PhD?
If it's different in the School of Psychology or Informatics, but at least in the School of Life Sciences, you have, so in your first year, you have 2 assessments. So you have your thesis committee, which is your co-supervisor plus 2 more researchers in the department. And you give like a 15-minute presentation, you write a really short report, which like gives an overview of the project. It's like very like friendly, you know, they're trying to give you support, help guide you in your project, and it's also like a well-being check-in as well, which is obviously very important during your PhD. And then you have another similar thesis committee that called later on in the year, and then after that it's just one every year. And then obviously at the end you have your final thesis submission and your viva, which is so that's when you have external examiners that come in, they go through your thesis, they go through your presentation, work out if you've got any gaps in your thesis that need plugging. So that's for life sciences.
In psychology, we don't have many assessments. We have something similar each year, where they review your progress throughout the year, and you might have to write a piece of work related to your research. And there's other than this, there's events like presenting posters and things like that, but these are not really assessments, more like opportunities for you to practice some important skills in communicating your research. Yeah, but not many assessments, I would say.
I feel like it's quite light on the assessments. You have the vibe, of course. And
Yeah.
Yeah.
Well, yes.
Yeah, I think informatics is the same. So you have your thesis committee where you have your second supervisor and somebody else, and so I thought I've got a better get hyped up for this and prepare all my stuff. And actually, it was an hour of what have you been up to? Yeah, that's fine.
Yeah. Are you okay?
Yeah,
yeah.
and it does feel to me you could always get to 3 years in, and then people find out you haven't been doing it. It does feel very assessment-like. I mean, as be a supervisor, I guess, is therefore, but it's more. It's almost. I feel it's up to you how much contact you have with your supervisor as well.
Yeah.
You could be hiding in the corner. You would know. I think.
Yeah.
Well, you do have to record meetings. But
Yeah.
it's not.
That depends how much of a stickler your supervisor is for. Once a year goes through and ticks everything that I've put in the meetings I've had. You're meant to have a meeting with your supervisor at least once a month. Yeah, and you record that.
I think that's another very important point to consider when you're thinking about doing your PhD is who's going to be your main supervisor. Because I think the relationship that you have with your supervisor is going to make or break your PhD. So I think it's very important to speak to people. If you're interested in a lab, it's very important to go and speak to members of that lab and also members of other labs which might be closely associated. They might be able to give you a more honest appraisal of what's going on in that lab. So yeah, I think it's just speak to people and find out what's good. And also different styles, something might work for one person and it doesn't work for another. My supervisor, I have a meeting with him every single week, and I love that, but I know for other people they would hate that. So it's what kind of would work for you as well, but that's very, very important.
Yeah, I agree. I mean, I can think of 2 supervisors in our department. One of them's mine, one of them isn't. And one of them will be, come back in 3 months and tell me how you've got on, and the other is weekly meetings. Right, this week you're going to do this, and you're going to report back to me next Friday with this. I don't know which I know which one I've got, and I don't know which would be more suitable for me actually. But so that's sort of don't just ask, are they a nice person or not, but what is their management style?
So someone is asking, Vanya is asking, how do you manage your time usually between classes, rotations, like that? Or I guess.
Yeah, not very well.
It's a learning progress, isn't it?
Yeah, I as I said, the nature of mine is investigating a lot of stuff, so you really have to be self-disciplined and say, well, I'm going to spend this amount of time in this, and if I haven't made any progress, I'm either going to decide to keep going or I'm going to ditch it. But I'm not just going to drift. You have to think that way. So that's managing what you do in your PhD. Then there'll be the odd other pressure on your time, like how much teaching you might do. Actually, I really like the teaching, so I tend to do that. It does keep you sharp as well doing that. And I feel, it depends on your nature. I mean, I try and treat it like a 9-to-5 job.
I was literally just about to say the same thing. I think that's the only way that I've gotten through is by doing it as a 9-to-5.
Yeah.
Because, again, it depends on the individual, but I think the second you let things slip, the days very quickly turn into weeks, turn into months, and then you're 6 months delayed on your experiments. So I don't know, my Google Calendar is very, very, very organised, and I just use that to organise my time. And I think there's lots of tools for time management and that sort of thing. But and also talking to your supervisor about how to prioritise, what to prioritise as well. But I think it comes with time.
I agree. Do you guys use to-do lists?
I've spent my whole life to do this, decided to waste of time.
I like to write out each Monday what are the bigger goals that I want to achieve that week. And I also realized that I used to put too many things, and actually you need to choose a few things that you think are realistic for you to achieve that week. And then it's very satisfying to tick them off.
Yeah. The satisfaction.
Yeah, because actually having a to-do list with 100 things on it is the most depressing thing ever. But if you get a to-do list with 2 things on it, if you can tick one thing, it can make you feel so much better about your day or your week or your month.
Keeps you motivated.
Even if it's just a simple thing, you manage to tick something off. It's good.
Questions from you guys?
I would like to ask about the research assistant job that you had before that would help you to decide whether you want to do a PhD.
So when I was doing the research assistant job, it was with the expectation, so I did start that in September with the expectation that my PhD would start the following September, so I was sort of already kind of committed to doing a PhD. But I found it very, very useful because it gave me a year to kind of faff around in the lab, understand, dip in and out of kind of things that I hadn't tried before. So I tried a bunch of 3D printing, just so that, because some people were doing that in the lab, so I tried doing that for a little bit. I like learning, spending time learning to code and learning how to use the two-photon microscope, the animal model that I use. So it was a very, very nice run-up to the PhD for sure, and it's meant that when I started my PhD, I was already sort of several steps ahead in the right direction to then start, collecting that proper data. So that job was on. This is being recorded. This isn't actually. So it was agreed with my supervisor that that's how it would sort of work. So it was the result of a conversation. That job.
I guess this shows that connections are very important in general in science.
Yeah.
Go to networking events. Come to these kind of things. This is really.
Yeah, I suppose your supervisor, Leon, so you've known him for a year as research assistant.
Well,
Did you know him before
Tom.
that?
It was with Tom actually.
Tom.
Yeah, and also I didn't know Tom before, Tom Bardo, but I knew Leon from my undergraduate. So I did my undergraduate and my master's here as well. And so I knew of him from then. But I think he sort of recognized me as well.
Yeah, so my supervisor, I taught, a course that I was very keen on, very heavily involved in my master's. So I'd known him for a year before I started the PhD. And been talking, in fact, he supervised my dissertation in my master's because I went to evidences there. So it's the same point you're making that, knowing people in advance smooths your way into it and helps you decide whether it's the right place to be or not. So get to know, if you're studying here now and you want to do a PhD here, think about who the supervisors might be and talk to them now. It might be 2 years before you're doing a PhD with them.
Sorry, just I was going to say, there's loads of opportunities for the summer internship stuff, and it's called JRA or something. You can just do a very short stint in someone's lab. And if it's possible for you as well, you could even do a week of unpaid work experience. I know that's not ideal, but it will also stand in good stead for that.
Well, the deadline for the JRA is Monday, so if you're interested in that, you better get looking. That can give you advice if you're interested.
Okay, very quickly because we're running out of time. If you have one short piece of advice for undergrads or graduates into getting into a PhD, what would that be?
When you turn up for an interview, you know what you're talking about. So I've researched what. So in my case, it wasn't just one supervisor I was talking to. It was a program of a committee. And find out who they are, find out what they do, so that you understand where they're coming from, and mold your answers accordingly.
I think it's good if you gain some research-related experience before for you to understand whether you would like this type of work, because the PhD is quite intense, and if you don't enjoy it, then I think it could be quite a miserable time. And so if you have a bit of understanding before that you would like this type of work, then that's a good start to decide that you want to do a PhD.
I think mine would be like what we said earlier, just like do your research on who your supervisors and
How they're like as a person.
exactly.
Yeah, great. All right, thank you everyone. Thanks to the panelists.